Every living thing needs to consume nutrition to function. Plants do this in a way different from us through the process of photosynthesis. The process uses energy from the sun to combine 6 molecules of carbon dioxide and 6 molecules of water to create a molecular glucose, the source of nutrition, and 6 molecular oxygen. Photosynthesis occurs in the chloroplast of a plant cell. Inside of the chloroplast, there are thylakoids and stacks called granums, with the surrounding area called stroma. The thylakoid is where the first stage of photosynthesis occurs, the light-dependent reaction. The chlorophyll inside the thylakoid membrane absorbs the light energy. The electrons in the chlorophyll are charged by the energy and bounce around through the chlorophyll. The chlorophyll is not able to contain the charged electron bouncing around, so it's passed from chlorophyll to chlorophyll until it reaches the last chlorophyll. The moving electron releases energy as it went through the chlorophyll. That energy is absorbed by an energy carrier ADP and becomes ATP. When the electron finishes going throughout all of the chlorophyll, an electron carrier NADP plus absorbs it, becoming NADPH. Thus, the setup for the second process of photosynthesis is complete and the light-independent reaction can begin.
The light-independent reaction takes place in the stroma and is the part of the process in which glucose is actually created. The water molecules are broken up and the bonds breaking release electrons, and those electrons go back to repeat the light-dependent reaction. The broken up water molecules eventually form oxygen with their leftover oxygen atoms. Within the stroma of the chloroplast, CO2 is combined with those hydrogen atoms in a chemical reaction with the help of the electrons from NADPH and the energy from ATP. As a result of the NADPH delivering its electron and the ATP delivering its energy, they are now NAD plus and ADP. Glucose has now been created with the byproduct of 6 oxygen molecules.
Many pesticides have been created to kill off unwanted plants by inhibiting the photosynthesis process of those plants. One example is nitrile pesticides. They are a selective herbicide used on contact to control the growth of broadleaf weeds and grass crops. One nitrile pesticide is bromoxynil, which was created in 1963 by Fone Pauling. The pesticide is used by industrial farmers in field corn, popcorn, small grains, garlic, onions, grasses, etc. It is applied to the foliage of the weeds and the plant rapidly deteriorates. The pesticide negatively affects the light-dependent portion of photosynthesis by disrupting the cell membrane of the plant. It blocks the electrons from transporting within the membrane. As a result, the electron is confined to one part of the chlorophyll membrane without a way to be passed on. These diverted electrons cause internal deterioration to occur, and the membrane is eventually.
Cellular respiration is necessary to provide the energy ATP in cells for all living things to function. In a cell, cellular respiration occurs in the cytoplasm that surrounds the organelles in the cell, as well as the organelle mitochondria. The entire process utilizes energy and electrons from glucose and oxygen to create ATP and the byproduct CO2 and H2O.
The next stage of cellular respiration is the Krebs cycle. It occurs in the mitochondrium within the matrix, which is a space within the inner membrane. The pyruvate formed from glucose undergoes a chemical change to easily continue the process. Each pyruvate becomes acetyl-CoA. The process for breaking down acetyl-CoA is extremely complicated, but the main idea is that it creates 3 carbon dioxide molecules for each acetyl-CoA. The chemical changes of the molecules releases electrons, and after 3 NAD+, receive those electrons to become 3 NADH, 1 FAD+. Another electron carrier also absorbs electrons and becomes FADH2. The energy release is absorbed by and ATP to become an ATP. Since there are 2 pyruvates, there are twice as many products, so there are 2 ATP, 6 NADH, 2 FADH, and 6.
The last stage of cellular respiration is oxidative phosphorylation. It occurs in the mitochondria between the inner and outer membrane and the inner membrane space. An electron carrier, NADH or FADH2 in the matrix, will drop off an electron to the protein chains within the inner membrane. After giving its electron, it becomes NAD+ or FAD+. The electron gets passed around through each of the protein chains. As the electron is out of the last protein chain, it combines with the molecule of oxygen and two hydrogens to create H2O, water. As the electron was passing through, it attracted hydrogen ions. But the protein chains don't want the hydrogen, and they shoot it through to the inner membrane space. In each protein channel, hydrogen is passed through into the inner membrane space. This causes an imbalance of hydrogen ions in the matrix and the inner membrane, called a hydrogen gradient. The hydrogen wants to go from the high concentration to the lower concentration of hydrogen ions, so it goes through the ATP synthase, which requires kinetic energy to do so. The kinetic energy is converted into chemical energy. ADP absorbs that energy and becomes ATP. Since there are so many hydrogen ions that pass through the ATP synthase, this portion of cellular respiration produces the bulk of the ATP in cellular respiration. It produces around 30 to 36 ATPs. Overall, the entire process of cellular respiration produces 34 to 40 ATP that our cells will use to function.
There are occasionally things that can interfere with the cellular respiration process. One such inhibitor is rotenone poisoning.
Rotenone is derived from certain types of plants in the pea family. In 1965, a French botanist found the chemical component in the plant, and then in 1902, Japanese chemist Nagai Nagayoshi rediscovered the crystalline compound and named it rotenone. Rotenone is now a general-use pesticide used for insect control and for fish eradications. It is dangerous because rotenone interferes with cellular respiration during the oxidative phosphorylation process in the electron transport chain in the mitochondria. To be specific, it inhibits the transfer of electrons. This interferes with the electron carriers NADH and FADH during the creation of ATP because it is unable to pass off its electron, creating a backup of electrons with the mitochondrial matrix. This prevents how much oxygen is available in cellular respiration. Rotenone causes damage to DNA and parts of the mitochondria because of the tissue anoxia by stopping the maximum rate of oxygen consumption at the cellular level.